Hey everyone, this is Module 7 video number 4, and this is the last video covering prokaryotic gene regulation. I have a few topics to talk about in this video, and they're listed here. One is positive regulation of the Lac Operon. So far we've been strictly discussing negative regulation of the operon, but we'll talk about how it can be also positively regulated. And also, another topic related to the Lac Operon is what's called catabolite repression, and this occurs when glucose is present on the environment. Finally, we'll wrap up the video by talking about a very brief example of repressible regulation, which is the case for the tryptophan operon, or the trpR. All right, so let's start off with positive regulation of the Lac Operon. So all the discussion so far has been about negative regulation, and that's through the Lac repressor binding to the operator, and that's preventing RNA polymerase from binding to the promoter. So it's interfering with RNA polymerase binding and transcription. There is also something that positively regulates the Lac Operon. And this is a factor called CRP, also sometimes called CAP. CRP stands for cyclic AMP receptor protein. This protein binds to cyclic AMP, which is a nucleotide that has been cyclized by an enzyme. So the combination of these two, the protein CRP and the nucleotide cyclic AMP, can form a dimer, two of those, and this is going to bind to the Lac Operon promoter. This binding will stimulate, attract, stabilize RNA polymerase binding to the promoter. Okay, so this is doing the opposite of what the Lac repressor is doing. It's promoting binding of RNA polymerase and transcription. Let's look at a slide showing how that works. So I like to use regulation of the Lac Operon, use the analogy of car, and the car has to have both the gas on and the brakes off in order to go. Similar for the Lac Operon. So it needs positive regulation, that's through CRP, bound to cyclic AMP. And it needs negative regulation off. That means the Lac repressor is not bound. So that's a situation when lactose is present. That produces the inducer, which binds to the repressor, and the repressor no longer binds to the operator. The brakes are off. This is the positive regulation part. So there's an enzyme that converts ATP to the nucleotide cyclic AMP. So these little yellow dots are meant to represent cyclic AMP. Cyclic AMP binds to CRP protein. All right, when you have two of those, it forms a dimer, and this binds to the Lac Operon promoter. And then that is going to serve as an attractant to stabilize binding of RNA polymerase. And if you stabilize RNA polymerase binding to the promoter, it's going to bind more often, and it's going to transcribe the operon more often. Okay, so when both of these are present, cyclic AMP and CRP, they bind as a dimer, and they stabilize RNA polymerase binding, and that increases the rate of transcription. And this slide is just showing how the CRP cyclic AMP dimer actually comes in contact with the RNA polymerase, and that stabilizes the RNA polymerase. Right? RNA polymerase is floating around, and it can bump into the promoter, but this is going to make it more stable, and therefore give it a better shot of starting transcription. Right? So that's going to overall increase the rate of transcription of the Lac Operon. Okay? So that's the positive regulation. In order for the Lac Operon to be expressed at high levels, you need both the gas on, this cyclic AMP CRP binding to the promoter and stabilizing RNA polymerase binding, and you need the brakes off, the repressor not binding to the operator. Now, there's another feature of the Lac Operon that comes into play, and this also involves the positive regulator that we just discussed. And that feature is that E. coli can use a number of different simple sugars or even disaccharides to generate ATP. Lactose has been the one we've been discussing, but actually glucose is the preferred source. So if E. coli finds itself in a medium that has both lactose and glucose, it will use glucose first, and it won't activate the Lac Operon for breaking down lactose. How does that work? First of all, that's called catabolite repression. The catabolite is glucose, and it's repressing expression of the Lac Operon. Okay? And you can see in the presence of both glucose and lactose, the Lac Operon is not actively transcribed. So the way glucose represses expression of the Lac Operon is by this feature or activity of glucose. It is an actual inhibitor of the enzyme that produces cyclic AMP. That enzyme is called Adenylyl cyclase. This slide shows the pathway for cyclic AMP production. It starts with ATP, the enzyme adenylyl cyclase, cleaves off a diphosphate, and it forms a cyclic branch between the remaining phosphate group and one of the carbons on the pentose sugar. That's cyclic AMP. That's the binding partner for the positive regulator, CRP. When glucose is present, this enzyme is inhibited, and very little cyclic AMP is produced. So, if this enzyme is inhibited, you don't produce cyclic AMP, and there's no binding partner for CRP, and none of this occurs. And so you're lacking the positive regulation. You're lacking the gas. Even though the repressor is not bound to the operator, you're not able to transcribe the Lac Operon at high levels because you're lacking the positive regulation. Okay? So that's why in the presence of glucose and lactose, the Lac Operon is not induced to be expressed. This is the final slide I want to show about the Lac Operon, and this is showing the actual DNA sequence in 2 lines here. So this continues on the bottom down here. And this is illustrating the promoter shown in kind of yellowish here, and the operator shown by this span. It's not a very big sequence. The operator is only 25 or so nucleotides long, and the promoter itself slightly longer. This is showing the cyclic AMP-CRP binding site. That dimer is going to bind here, and it's going to pull in RNA polymerase to bind here. Notice that the RNA polymerase binding site continues here. It is overlapping the operator, and it's also overlapping the Lac repressor binding site shown in purple here. So if the repressor is bound, the RNA polymerase cannot bind to the promoter. Okay? Okay, I forgot. I do have one more slide here. This is the summary slide of the Lac Operon. And just a note about this slide, it uses CAP in place of CRP. So it's color-coded here, showing the CRP binding site right next door to the RNA polymerase binding site. Here is the operator, and that's also the repressor binding site. And that's right in register with the start of transcription site. And just downstream from that is the first protein coding gene, the LAC-Z gene. Here's the situation when both glucose and lactose are present in the media. Because lactose is present, the repressor does not bind to the operator. But because glucose is present, the positive regulator, CRP, does not bind either. So the brakes are off, but also the gas is off. So there's no transcription. Here's a situation when glucose is present and lactose is absent. In that situation, you have both the brakes on, the repressor is bound, and the gas is off. There's no positive regulator binding. Okay. Here's a situation in the absence of both glucose and lactose. The positive regulator can bind because glucose is absent. However, the brakes are on. The repressor is bound to the operator. And so there's no expression from the operon. So for these three situations, the operon is off for different reasons. Here's a situation when lactose is present and glucose is absent. So in the absence of glucose, the positive regulator is present and bound to the CRP site. RNA polymerase can also bind. It's being attracted by the positive regulator, and also the repressor is absent. So that assembles RNA polymerase on the promoter, and that sets up transcription at a high level from this operon, and the operon is turned on. All right, so that's a summary of the Lac Operon, which is regulated both by positive and negative regulation. Let's take a look at the other main way that genes are regulated, and that is by repressible regulation. And if you remember from the first video, repressible regulation is seen for genes that code enzymes that catalyze anabolic pathways, synthesis pathways, in which small precursor molecules are built up to a larger product molecule. An example of that is the tryptophan operon, which encodes 5 enzymes that are used in the biosynthetic pathway for the amino acid tryptophan. So tryptophan is the end product, and this is the larger product molecule compared to the smaller precursors or intermediate molecules. The amino acids are needed in protein synthesis, of course. So E. coli wants to make tryptophan available constantly because it's constantly using protein synthesis to generate proteins. All right, so in repressible enzyme pathways, the default state is that they're on, but they can be turned off or repressed if the end product is abundant. So if an E. coli is making a lot of tryptophan and it's very abundant, it doesn't need to make more, it's going to turn off these genes by repressible regulation. And then only when tryptophan levels get very low will this operon be turned on. The repression will be removed. Okay, so let's look at how this operon works. The trp operon also involves the trp repressor. Is similar to the repressor for the Lac Operon. This repressor is capable of binding to the trp operator. When the repressor is bound to the trp operator, then the expression is turned off. The repressor can also bind to the end product of the pathway, tryptophan. And so that's how this repressor is involved in this pathway. Let's look at the details. When tryptophan is absent, we want the operon to be expressed so that more tryptophan can be made, which is needed for protein synthesis that's constantly going on. Okay, so in the absence of tryptophan, the repressor is not able to bind to the operator, and expression of the operon can occur. Okay. This is how it works. Top of the panel here shows when tryptophan is present. When tryptophan is present, it binds to the repressor. So trp by this oval, orange oval, the repressor by this purple colored shape. Presence of tryptophan, it binds to the repressor, and the repressor changes shape from its unbound form, which allows it to bind to the operator. This blocks RNA polymerase from transcribing the operon. Okay? And that's okay because you don't need these enzymes, 5 different enzymes, for the synthesis of tryptophan because tryptophan is abundant. What happens when tryptophan is absent? When tryptophan is absent, then there's no tryptophan to bind to the repressor. And the repressor takes this shape. This shape is not able to bind to the operator. And so the operon is no longer blocked, no longer repressed. And the RNA polymerase can bind the promoter, can transcribe all of these coding regions. They can be translated to the enzymes to make more tryptophan. Okay? So like the Lac Operon, it also involves a repressor. In this case, when the repressor is bound to tryptophan, that's what allows binding to the operator. When it's not bound, when it's not bound to tryptophan, it takes a shape that can't bind to the operator. All right, so it is repressed in the presence of tryptophan. The default state is in the on state, and it's only repressed in the presence of abundant tryptophan. All right, I'm going to end this video here, and in the next video we'll start talking about gene regulation in eukaryotes. See ya.